to...
So today's topic is DNA structure and DNA replication. That would be sections 13.1 and 13.2 in your textbook. Let's see what it's all about. So the first thing you need to know that DNA is made of nucleotides. That's its smallest unit, and you remember that when we did macromolecules earlier this year, I hope. And it also forms what is known as a double helix, which is two strands of those nucleotides, and they sort of do a little twisting so that they're like a helix shape, not unlike that picture. First thing we have to do is figure out what is a nucleotide. And so a nucleotide consists of three parts. One part is a nitrogenous base. That means something that is made of nitrogen, one of those elements that we learned about back in macromolecules. And those bases can either be A, C, T, or G. Now those letters stand for names. A is adenine, C is cytosine, T is thymine, and G is guanine. Those bases are attached to a 5-carbon sugar called deoxyribose. That's where you get the D in DNA. And there's also a phosphate group that helps to link the DNA together. Okay. Got a little ahead. There it is. Now you can write it down. A stands for adenine, T stands for thymine, C is cytosine, G, guanine. And one of the important rules that was discovered is that A will always base pair with T. And since it's a double helix, how do they line up? So A is always going to go with T, C is always going to go with G. And you need to remember that rule. It's super important. Now, the bases can either be purines or pyrimidines. A purine consists of two nitrogenous bases connected to each other. Okay, two rings, if you will. And so there's two of them. You don't have to know which ones are purines for our purposes, but you need to know purines exist. Pyrimidine has a single ring. The way I remember pyrimidine is one ring, and purines two is that pyrimidine sounds like primary, which would be like, primary school first. So that might help you. Moving on. If you have DNA, remember the cell cycle, you're going to make a copy of all your DNA. How do you do that? That's DNA replication. And so this happens, if you recall, during the S phase of the cell cycle. And you need to separate the two strands first if you're going to make a copy. And so that is done with an enzyme called helicase. And it's going to unwind the double helix so that you can get in there and make a copy. The new strands are made basically by base pairing. So wherever there's an A on the strand that you're trying to copy, the enzyme DNA polymerase will put a T. And they just keep adding them with the enzyme's help until you get a complete new strand. And you'll end up, after you copy your double helix, you'll end up with two double helixes. And this is a diagram if you look at DNA replication. And so here we have our double-stranded DNA opening up. And you can see the bases being added. They're showing the new bases are in the gold color. The original strand, or the parent strand, is in green. And you have, this is your giant DNA polymerase enzyme that's helping to make the copy. You'll also notice that this one's being made in this direction, and this one's being made in that direction. They don't really show that. But, so, and that's the end of the story.